Rice, Oryza sativa, or African rice, Oryza glaberrima, is the most important cereal crop in the developing world and is the staple food of over half of the world's population. It is generally considered a semi-aquatic annual grass plant. As a cereal grain, it is the most widely consumed staple food for a large part of the world's human population, especially in Asia and the West Indies. In Africa, cultivars with tolerance to iron toxicity and heat constraints have also been developed and cultivated. Rice is now grown in over 100 countries on every continent except Antarctica. It is the grain with the second highest worldwide population after corn, according to the data for 2010. Since a large portion of maize crops are grown for purposes other than human consumption, rice is the most important grain with regard to human nutrition and caloric intake, providing more than 1/5 of the calories consumed worldwide by the human species.
Genetic description of rice. Rice, scientifically known as Oryza sativa, is a crucial staple food for over half of the globe's population. It belongs to the Poaceae family and is primarily cultivated in two main subspecies, Japonica and Indica. Each subspecies exhibit distinct morphological and psychological traits influenced by their domestication history and genetic makeup. Genetic structure of Oryza sativa. Oryza sativa is a diploid species with a chromosome count of 2n is equals to 2x equals 24, organized into 12 chromosomes. Its genome span approximately 430 megabase pair or MBP and is renowned for its amenability to genetic modification, making it a model of organism in cereal biology's study. Subspecies and diversity. Japonica, domesticated in Yangtze Valley around 9,000 years ago, characterized by short, sticky grain. Grown primarily in temperate regions and upland areas of Southeast Asia. Next, Indica, originated in Indian subcontinent approximately 4,500 to 8,500 years ago. Features long, non-sticky grains, typically cultivated in lowland tropical region. And lastly, we have Tropical Japonica, a broad grain variety adapted to tropical conditions. Includes cultivar like tinawon and unoy, primarily found in high elevation area of the Philippines. Genetic traits and breeding. Rice breeding has focuses on enhancing yield and resistance to environmental stresses. Significant genetic traits include dwarfing gene. The SD1 gene is pivotal for producing shorter plants that are more resilient to logging and can support a higher grain yields. Resistant genes. Various genes confirm resistance to pests and diseases, notably against rice blast fungus or Magnaporte grigia. K-resistant genes include PI1, PI54, and PETA, which regulate immune responses throughout the plant hormones like salicylic acid. Molecular breeding techniques. Recent advancements in molecular breeding have facilitated the identification of quantitative traits or LUCI or QTLS associated with yield and stress resistance. These techniques include marker-assisted selection or MAS, utilize genetic marker linked to describe traits to enhance breeding efficiency. Secondly, genome. Wide association studies or GWAS aids in understanding complex traits by analyzing genetic variation across diverse rice population. Genetic engineering innovations. Innovations such as golden rice have emerged from genetic engineering. Efforts aimed at improving nutritional content by introducing beta-carotene biosynthesis pathway into rice grain. This initiative addressed vitamin A deficiency in regions where rice is dietary staple. So in overall, the genetic framework of Oryza sativa is complex and integral to its cultivation and improvement. Understanding the genetics not only aids in breeding programs but also enhances food security through development of resilient varieties capable of thriving under changing environmental conditions.
Oryza sativa is classified within the following taxonomic hierarchy. Its kingdom is Plantae, phylum Tracheophyta, class Liliopsida, order Poales, its family is Poaceae,